This video is about an article titled Knockdown of WSF1, a fly homologue of Wolfram syndrome 1 in the nervous system increases susceptibility to age and stress-induced neuronal dysfunction and degeneration in Drosophila. Wolfram syndrome, or DID-MOAD, is an autosomal recessive genetic disorder that is rare and causes childhood diabetes insipidus and mellitus, optic atrophy, and deafness, as well as affects the central nervous system. The WSF1 gene is very active in cells and provides instructions on how to make the protein wolframin. Wolframin is located in the endoplasmic reticulum of the cell. The ER is responsible for transporting nutrients around the cell, and the malfunctioning of the endoplasmic reticulum is what causes Wolfram syndrome to occur. The purpose of this article was to study the neurological and behavioral effects with respect to age of Wolfram syndrome in Drosophila via reducing the expression or knocking down of the WSF1 gene. In Figure 3, 4 different experiments or tests are shown. The first, Figure 3A, was a comparison of the mRNA levels of a normal fly, or control group, and a fly with a WSF1 mutation. It was shown that the levels were more or less the same and did not have a large impact on the fly. The second experiment, Figure 3B, was a comparison of the number of amino acids in a healthy fly versus a fly that had a mimic insertion or a transposon in the last exon of the WSF1 gene. The number of amino acids decreased from 852 to 592, showing that it was flanked due to the mimic insertion. The third experiment, Figure 3C, observed the ability of the flies to climb the vials they were in. The study showed that the more time that passed, the flies were less able to climb and spent more time at the bottom of the vial, showing that they had behavioral deficits because they were degenerating neurologically. The final test in this figure, Figure 3D, was to view the effects of the mutation on the optic lobe of the flies. The mutated flies, after 50 days, had more spots of neurodegeneration. The bar graph shows these results numerically in percentages. From day 35 to day 50, there was a drastic increase in vacuole area percentage, confirming that there was neurodegeneration and it was indeed age-dependent. These experiments showed that when the WSF1 gene was mutated with mimic and piggyback, age-dependent neurodegeneration occurred, causing behavioral changes and a shorter lifespan in Drosophila. The results confirmed that mutations in the WSF1 gene of an organism with Wolfram syndrome did not increase susceptibility but was actually responsible for age-dependent neurodegeneration. This paper leads to the field because the study done in Drosophila can help scientists understand what is happening on a neurological level when a person is suffering from Wolfram syndrome. In addition, it could possibly help in creating ways to prolong life for people who have this disease, as well as aid with or suppress the symptoms and other health problems that come with Wolfram syndrome.